Good morning, everybody. Well, at least this morning here. It's the last lecture. And so I'm going to finish up talking a little bit about proteins today. On the course website, I posted a practice final. Maybe you'd want to take a look at that and just to practice your skills before next week's final. The logistics of the final will be similar to exam 3 and exam 4. I'll release the final exam at 8 a.m. the day that the final is to be given, and you'll have until 5 p.m. the following day to submit to Gradescope. All right, so that's going to be the final. The final is a cumulative exam of the entire semester. There is no stress placed on the most recent material. It's just a cumulative exam of the entire semester. So check out that sample final if you want to practice up. Okay, so it's the last lecture. So I'm going to talk a little bit more about proteins, and then that will wrap it up for the semester and for the year. So last time we were talking about laboratory synthesis of a protein. Protein is a amino acid polymer, and we pointed out that if you mix two amino acids together in a beaker in a lab, nothing will happen. So we have to use a lot of cunning in order to make a peptide bond. In the laboratory, this is something that nature does very, very easily because Mother Nature is using enzymes. But in the laboratory, if we're just trying to do stuff in a beaker, it's a lot more difficult. So mixing two amino acids together, nothing happens. We have to use cunning. And so today I want to show how it is exactly that you would make a peptide bond in the laboratory. Okay, so talking about making a protein in the chemistry lab and how exactly that would be done. Okay, so we start out with we'll call it our first amino acid. Oh, let's say we're trying to make the amino acid glycine, glycylalanine. And remember, we talked about how the primary sequence of the protein is written in this format. This is the N-terminus and this is the C-terminus. So all we're trying to do is put together a glycine and an alanine together to make a small protein. Okay, so there's our glycine. Right, there's our glycine, and this is at pH, at a low pH, something less than 2. All right, so now this would be reacted with some kind of alcohol. We talked about this last time, reacted with some kind of alcohol, and we get a esterification of that glycine. Water is the other product of the reaction. Remember, a carboxylic acid and an ester, they're about the same reactivity. So in this acyl substitution, we are neither really going up the ladder or down the ladder. We're staying the same, so that's fine. Okay, so now what we would do is adjust the pH to something greater than 9, and the pKa of these ammonium protons, they're all about 9, so that when we raise the pH above 9, we're going to get deprotonation here. So we get this species. The pH doesn't affect this ester. Okay, and so now what we've done is we brought the nucleophile. We brought a nucleophile into play. This nitrogen atom has an unshared pair of electrons that can function as a nucleophile. And so now here's the trick, right? Here's some of the cunning that we have to use to make a protein in the laboratory. This guy, that's glycine, that's our so-called first amino acid, is now reacted with this anhydride, and it's going to be another acyl substitution reaction where there's the carbonyl carbon bonded to something that's not carbon and not hydrogen. This is going to go away, and the nucleophile minus a proton is going to take its place. So the adjustment of the pH to 9 was to allow this amino acid to react with this anhydride. Remember, these anhydrides are very high on the reactivity ladder, so that this reaction should pose no problem. Okay, so we get an amide, right? We've created, there should be a hydrogen here. We've created an amide from an anhydride. We're going down the reactivity ladder, so everything is cool so far. Okay, so did all of this stuff to the glycine, and now it's time to get the alanine into the act, and that way make our peptide bond. So we're still at pH 9, we're still at a high pH, so the alanine is going to come in with the NH2 deprotonated and the carboxylate. Right, there's our alanine at the pH that we're working at, which is pH 9. The carboxylic acid proton, that's gone, that fell off at around pH 2, and we're here at pH 9. And so now what we've set up is for another acyl substitution reaction where there's the carbonyl carbon bonded to something that's not carbon and not hydrogen. This bond is going to break, and the nucleophile, which will be this nitrogen, will take its place. So the tying up this nitrogen atom over here, that has solved a lot of the problems that we encountered when we talked about this the last time. We don't get self-reaction. This doesn't react with itself. We don't get that cyclization reaction. So everything here has been, all of our previous problems, or a lot of them anyway, have been solved by this procedure. All right, and so now this OR group, that breaks off, that's the leading group. This is the nucleophile that comes in, minus a proton, and voila, what we have done is to create that precious peptide bond, right, which is what you call the amide bond in a protein or a peptide. So we've created that peptide bond, we've stuck together the glycine with the alanine. Okay, and so now the last step of our laboratory synthesis is that now to remove this group, which has been a protecting group all along, so that this nitrogen can't be a nucleophile, so this nitrogen can't react with another molecule of itself or with the second amino acid that we made, so that this nitrogen can't do cyclization. We talked about all of the bad things that can happen in the procedure. All right, and so now the reagent that is used here, this is trifluoroacetic acid. Trifluoroacetic acid, and we're down at a pH of less than 2. Well, this is a carboxylic acid, it's acidic. It lowers the pH, and we run the reaction at this low pH anyway. And so what this reagent does is it selectively breaks up this amide bond, but it doesn't break up the peptide bond. So this is kind of surprising chemistry, but this trifluoroacetic acid is selective for removing the amide bond from this group here. Okay, and so we're at low pH. What we've done is liberate the N-terminus. And there's our peptide. Here's our glyallopeptide that we were trying to make in the first place. So you see there's a lot of the laboratory synthesis of a protein. There's a lot of rigmarole involved because when you add two amino acids together in a beaker, absolutely nothing happens. So in order to make the peptide bond, in order to make this protein, this small protein, how many steps? One. Okay, this is just adjusting the pH. That's not really any chemistry. You're just adding OH minus. 1, 2, 3, 4. 4 steps. That's quite a lot of steps. If you can imagine that, let's say that every step has a 50% yield, right? Because when you run a reaction in a laboratory, you frequently don't get 100% yield. So let's just say maybe 50% yield for each of these steps, and we had 4 of them. So what would be the overall yield? It would be 1 over 2 to the 4th power, right? We're talking just about a few percent yield of this peptide using this procedure. And if we wanted to stick on another amino acid here, we'd have to go through this all of this rigmarole once again with protection there, esterification there, reaction with this anhydride, and then we would stick on the 3rd amino acid. 4 more steps, right? We're up to 8 steps. And then if we wanted to stick another amino acid on, it would be a total of 12 steps. So obviously, making a protein in a laboratory is a very involved procedure, but it can be done. And in fact, all of this chemistry was deemed worthy of a Nobel Prize for Professor Robert Merrifield of NYU, who cooked up this scheme in the 1970s. And laboratory synthesis of the proteins has come a long way since the 1970s. This procedure is now totally automated, right? You get a get a machine. You buy the machine. The protein synthesizer costs about $250,000. And you fill up a bunch of reservoirs with all of the amino acids that you want to string together. You push a button, and the test tube that has the first amino acid gets esterified, and then the pH is adjusted. Then you push another button, and this anhydride is delivered to that test tube, and then you push another button, and then the second amino acid is delivered, and then you push another button, and this trifluoroacetic acid is delivered to the test tube. And then you do all of this button pushing again. And even though the procedure is automated, and even though it's not someone necessarily in the lab fiddling around with test tubes and heating things on a heating mantle or a boiling water bath or stuff like that, because there are so many steps, protein synthesis of a big protein that has more than maybe 8 amino acids in it, it's just not practical in the laboratory, but it can be done. And so if you're interested in making small peptides, this is how you would do it using an automatic amino acid analyzer. An automated protein synthesizer. Okay. Now, next topic I want to get into is something that is kind of interesting and timely, in that it is the sequencing of a protein. We talked about how a protein has many levels of organization, many levels of structure. There's the primary structure, which is the sequence of amino acids. Then there's the secondary structure, which is that helical regions and the beta pleated sheet. And then there's the tertiary structure where amino acids that are separated by big distances in the primary sequence are brought close together by disulfide bonds, hydrogen bonds, and other interactions. And there are even more levels of organization. Okay, so what I want to talk about is how you can determine the primary sequence of a protein. Which amino acid is stuck to which amino acid is the N terminus, which is the C terminus, and so on and so forth. Okay, so let's talk about protein sequencing. But before I get into that, let me ask you something. Has anyone submitted a DNA sample to one of these commercial services that tell you what ethnic group you belong to and whether you have a propensity for this, that, and the other metabolic or genetically inherited disease? Maybe you 23andMe, I think, is one of the companies that does this, and there are many, many others. That is DNA sequencing, because the DNA molecule also has a primary structure that's telling you which nucleotide is bonded to which nucleotide from one end to the other end of the chain. So sequencing of biomolecules, proteins, and DNA, well, this is big, important science. It's an industry that is worth billions and billions and billions of dollars per year. All right, but we are in no position to talk about DNA sequencing. The tool, we can only talk about protein sequencing. The tools, the techniques, the methodology, all of that is all very similar. Okay, so let's talk about the tools. Protein sequencing. What is the primary structure of the protein that you might be interested in? Okay, so here are the tools. First of all, total hydrolysis. You get the protein and you react that with water under acidic conditions. You heat it, and the protein, because it's a polymer, amides, the protein is an amide polymer. And so we have all of these carbonyl carbons bonded to something that's not carbon, not hydrogen, bonded to nitrogen. The water takes the place of those leaving groups. So total hydrolysis of the protein takes the protein and just chops it up into the amino acids. Okay, so this tool, this tells you how many amino acids. I'm just going to abbreviate that AA, amino acids. This tells you how many amino acids are in the protein. It doesn't tell you its sequence. It just tells you how many they are and how many amino acids and their identity. So do you have valine in there? Do you have glutamine in there? Is it tyrosine? What? So total hydrolysis tells you how many amino acids and what they are. Okay. Then we have N-terminal analysis. This is called the Edman degradation. And so you feed the protein that you're trying to sequence to the Edman reagent. I'll show you a picture of what this is later on. And it tells you what the N-terminal amino acid is. This Edman reagent, it just chops off the first amino acid at the N terminus, and so now you know what is at the N terminus. Okay, the rest of the tools that are involved in protein sequencing all involve enzymes. And in DNA sequencing, like they do over there at 23andMe, it's the very same thing, is that they feed the DNA to enzymes, and the enzymes chop up the DNA at reliable, reproducible locations. And it's the same for a protein. You feed the protein to an enzyme, and the enzyme chops the protein up in a reliable, reproducible way at certain locations. Okay, so this is really the premier tool here of protein sequencing. You have these enzymes. One of the first one is carboxypeptidase. Okay, this is an enzyme, and it chops off, it bites off the C-terminal or last amino acid. This tells what the C-terminal amino acid is. Okay, the other enzymes that are used here, we have chymotrypsin. Chymotrypsin is a digestive enzyme. This is in your stomach. This is a protease, whose job it is to take that hamburger you're going to eat for lunch, or that tofu that you're going to eat for lunch, and it takes the protein in those foodstuffs and chops them up. And that's one of the steps in the digestion of food. So this is a digestive enzyme. And chymotrypsin breaks the protein chain after the arene amino acids. So that's the peptide chain. And what chymotrypsin does is it breaks the protein chain after a phenylalanine. It breaks it after a tryptophan, and it breaks the chain after a tyrosine. Right, so this wavy line that represents the rest of the amino acids in the primary sequence. You don't have to memorize any of this, right? On the final exam, I'll just give you this information. You do need to know how to work with it and what this representation means. Okay, another enzyme that we use is trypsin. This is another digestive enzyme. It's found in your stomach, and it takes that hamburger, it takes that tofu, and it starts breaking those protein molecules down into amino acids. Trypsin breaks the protein chain after a lysine residue and after an arginine residue. Okay, and so now, fortunately, I think we're going to have room here. Now there are a couple more enzymes here that are important. There's something called staph V8. It comes from they isolated this enzyme from the bacteria, from Staphylococcus bacteria. That's what the staph means. And staph V8 breaks the protein chain after an aspartic acid residue and after a glutamic acid residue. So this enzyme specializes in breaking the chain after the acidic amino acids, aspartic acid and glutamic acid. Finally, we have endopeptidase. And this is another digestive enzyme. It's a little bit different from the other enzymes that we've talked about because chymotrypsin, trypsin, and staph V8 break after an amino acid. This guy breaks the chain before on the N-terminal side. So endopeptidase breaks the chain before an aspartic acid residue, and it breaks the chain before a cysteine residue. Okay, so these are the tools that are used in analysis of the primary sequence. Okay, so let me show you some pictures, and then we can do some problems sequencing a protein. Okay, let's see. What do I want to do now? Share. Okay, so here's one of those ribbon diagrams of trypsin. The molecular weight of trypsin is about 23,000 grams per mole. That's a huge molecular weight. It's made up of 200 amino acids. And so we talked about how amino acids have helical regions and how we have beta pleated, and that's represented by these arrows, the helical region by these corkscrews. And then we have regions of the protein that are neither helical nor beta pleated. That's just this this filament here, but this is a bunch of amino acids all strung together. Okay, so that's trypsin. That's trypsinogen. Forget about that. This trypsinogen, this is kind of interesting because, you know, trypsin breaks up proteins. All of your soft tissues are made out of protein. So that if you had all of the trypsin in your stomach that you need at any given time, the trypsin would begin eating holes in your stomach because your stomach is made out of protein. So there's very little trypsin in your stomach or anywhere until it is called upon by biofeedback mechanisms to be released from the pancreas into your stomach. What you have a lot of is this thing trypsinogen, which is the pre-enzyme. This guy doesn't break apart protein, right? It undergoes a reaction in which trypsin is created from trypsinogen. So this way you don't get holes eaten in your pancreas and in your liver and in your stomach because there's very little trypsin around at any given time until there's food in your stomach, and then trypsin goes to work. Trypsin is created from trypsinogen, which is how trypsin is stored when it's not needed. Okay, here's a picture of chymotrypsin, another ribbon diagram of this enzyme chymotrypsin. It's about 240 amino acids. And once again, there are helical regions and there are regions of beta pleating. And this says catalytic triad. This is where all the action is in the chymotrypsin, where this catalytic triad is. It's 3 amino acids, a serine residue, a histidine residue, and an aspartic acid residue. And this is where the actual digestion or breaking apart of a protein actually happens in this active site where the catalytic triad is. Okay, I talked about the Edman reaction and the Edman degradation. And so here I have this slide that just pictures the chemistry. I don't expect you to know what the Edman reaction is telling you about a protein, but I don't expect you to know anything more than that because this chemistry is all very, very complicated. This is graduate school stuff. So there's the Edman reagent, and here are the first 2 amino acids of a protein. And what the Edman reagent does is it bites off the first amino acid or the N-terminal amino acid. So I've highlighted the N-terminal amino acid in blue. It reacts with the Edman reagent at pH 9, and then there's a hydrolysis, and the final product is this thing, which has the remnant of the first amino acid in it. So this isn't an amino acid, but it contains the remnant of that first amino acid. And the analytical chemistry of this thing is very well known, so it's easy to determine what exactly that first amino acid is. That's what the Edman reagent does. It bites off the first amino acid. Okay. Okay, so let's sequence a protein. All right, so I'm going to leave all of this info up here just for the record. It may take up too much board space. I may have to erase it. All right, so these puzzles are kind of fun, or at least I think they are anyway. Okay, so we have a protein. And we subject it to total hydrolysis. Subject the protein to total hydrolysis. And remember, this is telling us how many and what the amino acids are. And so in this unknown protein, we got, this is listed in alphabetical order. We've got 3 glutamic acids, a phenylalanine, a proline, 2 serines, 2 threonines, a tyrosine, and 2 valines. Okay, so those are the amino acids in the peptide. Okay, we took the Edman reagent, and the Edman reagent liberated proline, right? So this is telling us what our first amino acid is. Okay, and so then we took staph V8. We took staph V8 and we got 3 fragments because staph V8 breaks after an aspartic acid and after a glutamic acid. We got 3 fragments. I'm going to call them S1, S2, and S3, the S standing for staph. So we got S1, and S1 was not analyzed. We didn't have to. It's not analyzed. So that's a big mystery. But the fragment S2 turned out to be glutamic acid, 2 threonines, and a tyrosine. And then we took the fragment S3, and that was analyzed. That turned out to be a glutamic acid, a phenylalanine, a proline, a serine, and 2 valines. Okay, finally, we took chymotrypsin, and once again, we got 3 fragments. I'm going to call them CH1, CH2, and CH3. These are the 3 fragments that we got when the protein was digested with chymotrypsin. CH1, once again, was not analyzed. Just to save time. CH2 turned out to be a phenylalanine, a proline, and a serine, and CH3 was a glutamic acid, 2 threonines, a tyrosine, and 2 valines. Okay, I'm going to have to erase this, but let's just maybe up here. Chymotrypsin breaks after a phenylalanine, after a tryptophan, and after a tyrosine. And staph V8 breaks after an aspartic acid and after a glutamic acid. Okay, so ran out of room here. Okay, so there's all of the information that we're getting, and now we can sequence the protein. Okay, so here's what we do. First of all, set up a scorecard with one line for each amino acid. So this is telling me on my little scorecard here, I need 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 amino acids. So I'm going to put 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Little hash marks there, and that indicates the 12 amino acids that I'm going to find a home for in the primary sequence. Okay, now we can start using some of this data. All right, so the Edmond is telling us that the first amino acid, the N-terminal amino acid, was proline. Right, that's what the Edmond degradation does. So we found a home for proline. Okay, and now the rest of the analysis involves finding where on this peptide these fragments were. And we want to mark out where these fragments were. Okay, so I'm looking at this and I see there's only one proline. And the proline came out in this fragment called S3. See, there's the proline. There's only one proline. He's unique. So this guy was in S3. S3 is 1, 2, 3, 4, 5, 6 amino acids long. So on my little scorecard here, I'm going to mark out 6 amino acids. 1, 2, 3, 4, 5, 6. There's my fragment S3. Right, I'm taking 1, 2, 3, 4, 5, 6 amino acids, and now I know where S3 is. Okay, another thing that I know is that staph V8 breaks after an aspartic acid and after a glutamic acid. Well, I don't have any aspartic acid here, but I do have glutamic acid, so that this guy, the last amino acid in S3, had to be a glutamic acid because staph V8 breaks after a glutamic acid residue. Okay, so now I'm going to look at the rest of this data, and I'm going to do the very same thing. I'm going to find on this peptide where exactly all of these fragments live. Okay, here's that unique proline residue, of which I only have one, and it came out in this fragment called CH2. And CH2 is 3 amino acids long, so CH2 must be down here. There's the fragment CH2. And what else I know about CH2 is that it's a chymotrypsin fragment, and chymotrypsin breaks after phenylalanine, tryptophan, and tyrosine. So CH2 has this phenylalanine in it. That must be where the phenylalanine lived, because chymotrypsin broke after this phenylalanine residue. So PHE has to live there. That's where the chymotrypsin broke. All right, and so CH2 is phenylalanine. I found a home for him. And it's a serine, so serine has to live there. I'm just using what was in CH2 to determine where the serine was. And now if I go back to S3, I see that S3 overlaps with CH2. I see in CH2, I see phenylalanine, proline, and serine. Phenylalanine, proline, and serine. So S3 and CH2 overlap. I found a home for him, for him, for him, and for him in S3. The only thing I'm missing are these 2 valine. So they must live there. So you see the technique? The technique is finding where these fragments are in the peptide, and you're looking for regions of overlap. Okay, now, oh, see, now I found where these two valines live. And those two valines came out in CH3. CH3 is 1, 2, 3, 4, 5, 6 amino acids long. So CH3 has these two valines and it's 6 amino acids long. 1, 2, 3, 4, 5, 6. So that's where CH3 must have been. And this is where CH1 has to be, right? Which we don't have any information about yet. Okay, so let's go back to CH3. CH3 is a chymotrypsin fragment. It breaks after PHE, TRP, and TYR. We see that CH3 has a TYR in it, a tyrosine. And since chymotrypsin breaks after a tyrosine, then the tyrosine had to live there. Okay, CH3. We found a home for glutamic acid. We found a home for tyrosine. We found a home for two valines. Now we need to find a home for these two threonines. We have two empty slots here, so that must be where the threonines are. Okay, so we're doing pretty good here, right? Okay, so now two threonines, huh? Oh, the two threonines came out in this fragment S2. Here they are. S2 is 1, 2, 3, 4 amino acids long. So this must be S2. There's S2. And staph V8 breaks after a glutamic acid. And so the last amino acid in S2 must be this glutamic acid. Okay, so how we do it? Well, we found a home for these two valines. We found a home for the tyrosine. We found a home for these two threonines. We found a home for one serine, but not the other one. And we found a home for two of these glutamic acids, but not the third one. So what we're still looking for is where is that serine and where is the missing glutamic acid? So because staph V8 breaks after glutamic acid can't be here, right? We're trying to find a home for GLU and we're trying to find a home for SER. GLU can't be here because staph V8 would have broken after that glutamic acid residue. So the only thing that makes sense is serine there and the glutamic acid there. So we were looking for the missing serine. We were looking for the missing glutamic acid. This must be the fragment S1. So there it is. We've sequenced the peptide, right? And so we're integrating all of this data, right? We can't solve the problem by analyzing all of the S fragments and then analyzing all of the CH fragments. No. We have to find regions of overlap and we have to be bouncing back and forth between all of the data that we have. Okay, so this was a dodecapeptide of 12 amino acids. We could sequence it using pencil and paper, but something like chymotrypsin, that has 250 amino acids. Imagine trying to sequence chymotrypsin, on a blackboard up here using pencil and paper. Have you guys ever heard of the field of study called bioinformatics? This is what bioinformatics does, is that when you get a big protein or a huge DNA molecule and you feed it to enzymes, you're going to get hundreds, sometimes even thousands of fragments. Here we only got 6 fragments, right? But sometimes if it's a big protein or a or a DNA molecule, you're going to get hundreds, thousands of fragments. How do you find the regions of overlap the way we did it here? That's bioinformatics. You bring computer power to work. And so the computer figures out, people program the computer to find these regions of overlap, to find these empty spaces. And so that is how DNA is sequenced. Have to use a computer to sequence something that's really big. So how did that go? Fun, huh? Sherlock Holmes, huh? Okay, let's try another. Okay, so let's just try for fun and practice sequencing another protein. And here we've got a protein, and total hydrolysis gave us 2 alanines plus 2 aspartic acids plus 2 glycines plus an isoleucine plus a phenylalanine plus a serine plus a tyrosine and then 2 valines. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So we're going to set up our little scorecard here of 12 blocks. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So that's what total hydrolysis is telling us. Edmund liberated serine. All right, and Edmund is telling you what the N-terminal amino acid is, so we can immediately write serine there. We found a home for serine. Carboxypeptidase, that liberated isoleucine. And remember, carboxypeptidase, that is telling you what the last amino acid is, what the C-terminal amino acid is, so we can immediately write isoleucine at the end there, so we found a home for him. Okay, now for the enzymes. Chymotrypsin, we got 3 fragments. CH1 worked out to be 2 alanines, an aspartic acid, and an isoleucine. CH2 was not analyzed, didn't have to, could figure out the primary sequence without it. And CH3 turned out to be 2 glycines, a phenylalanine, and a serine. Okay, and remember chymotrypsin. Chymotrypsin breaks after PHE, TRP, and TYR. Okay. Then they used endopeptidase. And once again, 3 fragments were isolated. I'm going to call them E1, E2, and E3. E1 was not analyzed. E2 turned out to be the same, exactly the same thing as CH1. That was convenient. And the fragment E3 consisted of alanine, aspartic acid, and isoleucine. And so just for the record, let's remember that endopeptidase breaks before an aspartic acid and before a cysteine, right? Some of these other enzymes break after, but endopeptidase breaks before. Okay, so do you think you could do this problem? Do you think you could sequence it? What are we looking for? We're looking for regions of overlap. Okay, so there's only one serine. We know where he lives at the front. And so we see that serine came out in the fragment CH3. And CH3 is 1, 2, 3, 4 amino acids long. So this must be CH3 here. Okay, and it's a chymotrypsin fragment. Chymotrypsin breaks after the arene amino acids. And so in CH3, we have a phenylalanine, and chymotrypsin breaks after phenylalanine. So phenylalanine has to live there. And furthermore, CH3 has 2 glycines. We have 2 open spaces here in CH3. That must be where those 2 glycines live. Okay, so that was CH3. All right. Oh, CH1 has the isoleucine in it, right? And there's only one isoleucine, and we know where he lives. So this fragment CH1 is 1, 2, 3, 4 amino acids long. 1, 2, 3, 4 amino acids long. This must be the fragment CH1. And this must be the fragment CH. This is CH1. This must be the fragment CH2, which we don't have any information about, except that we know that it's a chymotrypsin fragment. And I'm looking at this guy here, and I see chymotrypsin breaks after phenylalanine. We only have one. We know where he lives. And chymotrypsin breaks after TYR. We have one TYR. We don't know where he lives, but since chymotrypsin breaks after TYR has to be there. Okay, so we found a home for him. Okay, so now let's jump over here to the endopeptidase data. The fragment E2 was the same as CH1, so this is E2 down here. And something's wrong. Oh, shoot. E2 E2 is the same, oh, sorry. E2 is the same as CH3. Sorry about that. Just wrote down the wrong number. So E2 is the same as CH3, so E2 is there. Okay, so I think now we're back in track. I just wrote down the wrong number here. It's supposed to be the same as CH3. Okay, so now what? Okay, so now this fragment E3 has this isoleucine in it, and we know where isoleucine lives. E3 is 3 amino acids long, so E3 must be here. There's E3, and this guy here in the middle, that must be that must be E1. Okay, so how are we doing so far? Okay, so now let's see, what else can we do here? Is we know that endopeptidase breaks before an aspartic acid residue, and also before a cysteine, but we don't have any cysteine, we just have aspartic acid. So if we look at these endopeptidase fragments, there had to be an aspartic acid there because endopeptidase breaks before an aspartic acid, and that's where we had the break. And if we look down here, this guy here had to also be aspartic acid because endopeptidase breaks before an aspartic acid. Okay, so now what else can we say? E3 is alanine, aspartic acid, and isoleucine. We have one open slot in E3. We found a home for aspartic acid. We found a home for isoleucine. We have an open spot for alanine, so alanine has to be there. And what else? CH1 has two alanines, an aspartic acid, and an isoleucine. So there's, we're in CH1, we're missing one alanine. So that must live there in CH1. Okay, so we're doing great. We found a home for everything. Two alanines, check. Two aspartic acids, check. Two glycines, check. Isoleucine, check. Phenylalanine, check. Serine, check. TYR, check. We have two open slots. We haven't found a home for valine. So that must be, those valines have to live there. So how'd that go? Well, I hope. So if you look at the course website, I put a practice problem dealing with sequencing the peptides, and so you might want to take a look at that. I do so wish that this semester did not turn out the way it did, but you know, just stuff happens. I wish we were able to be together throughout the whole semester, but I guess it didn't work out that way. I do thank you very much for your attendance and for your attention, and I will be in touch by the Blackboard about the logistics and the final and office hours and all of this other stuff. So that's all I have for you today and for the semester. See you down the road. Any questions about anything before we sign off for the last time? Oh, thank you. It's my pleasure. Any questions or anything? Oh, yeah, no, thank you. Thank you for everything. Okay. Anything else? Oh, no, yeah, thank you. Thank you for your attendance and for your attention. All right, well, I'll see you down the road and I'll be in touch via the Blackboard website. Oh, thank you. Try to stay safe.